want to do a brief discussion on some medical techniques, not all of them obviously, but some medical techniques used in reproductive physiology and so forth. What I'd like to discuss is in vitro fertilization, which helps women who cannot get pregnant, somatic nuclear transfer, where you can clone an entire organism, therapeutic cloning tissue like to help with diabetes or so forth like that, induced pluripotential stem cells, and that changes an adult cell back to a pluripotential stem cell, and regenerative medicine I want to talk on. So let's just discuss that for a moment there, and we'll okay. So in vitro fertilization starts by a woman undergoing FSH hormone injections or analogs. Analogs would be something like Clomid. What the intent is to have the woman multiple ovulate, to ovulate a lot of oocytes, hyperstimulate the ovarian follicles, okay, because they need a bunch of them. The oocytes are then harvested, and they are taken out, in other words, harvested, and they're fertilized through really an injection of sperm into that oocyte, which would be in metaphase 2. They're then allowed to develop for 3 to 5 days and then transferred back to the uterus of the mother. And they implant several of these embryos because it takes several to get one viable, but however, sometimes more than one, so the lady may get multiple pregnancies. And the process has allowed millions otherwise infertile couples to have babies, okay? And so that's that in vitro fertilization aspect there. Here's the picture of in vitro fertilization where the sperm is injected into this oocyte here to show you on this in vitro fertilization aspect. Okay. Now go to something else, introduce a concept called genomic equivalence. Remember, a genome is a complete set of genes in an organism. So in a genomic equivalence, all the cells in the human body have all the genes, all the same genes, okay? Of course, the sperm and egg have half as much, but they still have a complete set of genes. That's what we call genomic equivalence. Differentiation involves closing or inactivating some of these genes, which basically means that a kidney cell and a liver cell all have the same genes, but some of the genes in the kidney cells are inactivated that the liver cell would use, and vice versa. So all of them have the same, but differentiation involves inactivation of genes, which can involve zinc fingers, methyl groups, whole various things that can do that. Okay. I want to introduce the types of stem cells. Okay. Stem cells can do differentiation, mitosis. They can reproduce themselves and do further differentiation. So they can do proliferation and differentiation. Proliferation and differentiation. The key is how much differentiation can they do? Again, going back to this genomic equivalence, closing genes and so forth. So a totipotential stem cell can differentiate into any cell of the organism, including the extraembryonic tissues like the placenta, the allantois, the yolk cell, all those things. A pluripotential stem cell can differentiate into all the cell types of the body itself, but not the extraembryonic structures. So it is said that once you get up to the 8-cell stage, after you start doing the eight blastomeres, after you start doing cleavage, they would be totipotential. So if I took one of those, any one of those could develop anything for the whole organism. Okay. Multipotential stem cells are really like progenitor cells. They can differentiate into discrete cell types, like different types of blood cells and so forth. Okay. All right. So those are just some terminology. So I'm going to go here. So a stem cell in a multicellular organism, stem cells are undifferentiated or partially differentiated, we talked about. They can differentiate into various cell types and proliferate indefinitely to produce more of the same stem cells. They are the earliest type of cell in the cell lineage. They are found in both embryonic and adult organisms. Okay, adult stem cells we'll talk about, but they have slightly different properties in each. They are usually distinguished from progenitor cells, which cannot divide indefinitely. So when we go back here, see, they cannot divide indefinitely, which would be more like the multipotential, like the blood cells. Okay, so we come here, coming to a concept called somatic nuclear transfer. This is what they call the cloning. Like you could make a whole new sheep, dog. They've done this. Basically, cells of early cleavage up to 8 cell stage are totipotential, as we mentioned. A cloned blastocyst can be created by implanting a mature fetal donor skin cell or cell of the somatic cell nucleus into the ovum cytoplasm of the somatic cell. Okay, let's just kind of look at what I'm saying. All right. This sheep here donates an egg, okay, with the nucleus. Okay. The nucleus is taken out. This is a mature cell. This is mature but still young. Because remember what I said, you have to get more like a fetal or baby young. You take the nucleus of this, put it into this cleaned out egg, I guess you could say the nucleus has been removed, and let it multiply. Because again, this right here is totipotential. So by virtue of being totipotential, it can make anything, which would be a new offspring. You may want to study on that or look it up. Somatic nuclear transfer. That's cloning. That is cloning, should we say. Okay. Therapeutic cloning is you take some cells from the inner cell mass. Now remember, the inner cell mass has already done differentiation. That's where the person is going to come from. They are pluripotential. They cannot clone because they can't do the extra embryonic tissue that supports the development of the embryo. But you could make tissues from this. Tissues. You could take some cells from an individual, do this technique here, and make tissues. These cells cannot clone an entire new organism. Why? Okay. The answer is because there's already been some differentiation. There's already been some differentiation to form the trophoblastic cells that form the chorion and other extraembryonic structures. But they can be used therapeutically to make cells like the beta cells in the islet of Langerhans to help with patients with type 1 diabetes, regenerate neurons, spinal cord injury, and so forth. So here's stem cells again. Adult stem cells found in the hippocampus of the brain subventricular nucleus. You can see multipotent can give rise to several cell types. Though it's multipotent, they cannot jump across germ layers. So you can't take an ectodermal cell and make it a mesodermal cell. That's where this multipotent. But anything within the realm of, let's say, mesoderm, which can make connective tissue, and it can make muscle. So you theoretically could take a fibroblast and convert it muscle cell. Induced pluripotential stem cells. These are cells, adult fiber can be changed into pluripotent cells using retroviruses. So in other words, you take an adult cell like a fibroblast, okay? You use a retrovirus to introduce some certain genes. That's how we introduce genes into, and these genes would turn on transcription factors So other words, to make a long story short, you're taking an adult cell and converting it back to a pluripotential stem cell. That's why it's called induced pluripotential stem cells. Adult cell and convert it back by introducing certain genes by using viruses, and these genes code for transcription factors. Okay. Regenerative medicine is a new field of medicine that's emerging using various techniques, particularly the techniques of this here, which would be therapeutic replacement. Hopefully one day type 1 diabetes and spinal cord injuries could be alleviated because you could do various things to manipulate the cells to become those types of cells. So that's just a little introduction into that. I'm not going into a whole array, but this gives you sort of an idea.